Yes, so welcome to my YouTube channel. Don't forget to subscribe, like and share. Okay, so today we're talking about fertilization. So fertilization, as you know, is just a fusion of two highly specialized and differentiated haploid germ cells. The germ cell we're talking about is the oocyte and the spermatozoon. Okay, the spermatozoon from the male, the oocyte from the female. When they fuse together, they form what we call a zygote, and the zygote undergoes meiotic division. Okay, but when the two um, when the two haploid cells fuse together, that's the completion of the second meiotic division. The meiotic division we're talking about is just the cell division. Okay, so that's what we mean about fertilization. In simple terms, it's just the fusion of the male gamete, which is the spermatozoon, and the female gamete, which is the oocyte, to form a zygote. Okay, so fertilization, as said, it's just the fusion of male and female pronucleus. The nucleus of the ovum becomes the female pronucleus. So the head of the spermatozoon, which is formed from the nucleus, separates from the middle piece and the tail. Okay, that's how it happens. The spermatozoon, shades of its tail when it wants to penetrate the nucleus of the oocyte. So thereafter, the pronucleus uses the nuclear membrane, then the 23 chromosomes from the female pronucleus and from the male pronucleus. The pronucleus we're referring to the same, the oocyte and the spermatozoon. It's a pronucleus. Pro means before, because it hasn't become an active nucleus. So when they mixed up, they get to form a pair of 46 chromosomes, which is now a diploid, a zygote. Okay, so that's how these things happen. So you can see this is just a picture showing the sperms when they come to enter. As on 1, you can see the sperm approaches the egg. Then on 2, the sperm acrosome enzyme digests the enzyme code. Then on 3, protein of the sperm head binds to the receptors. That's the coronary radiator. That's where it binds. And then on 4, the plasma membrane of the sperm and the egg fuse. The sperm nucleus enters the egg cytoplasm. That's on 5. On 6, fertilization membrane forms. Okay, so those are the processes. You understand them later. Okay, so this is the picture of it. Try to take a minute, go through. Okay, so on 1, sperm makes contact with the egg. On 2, acrosome react with the zona pellucida. Okay, the zona pellucida, it's a membrane as well, which is after the first membrane. Okay, so it follows by the zona pellucida. After the coronary radiator, it follows by the zona pellucida. Okay, so the zona pellucida is the one that can only give access the spermatozoa to enter the membrane and find itself in the cytoplasm of the female oocyte. Okay, so on 3, as you can see, the acrosome reacts with the perifertile space. Okay, on 4, plasma membrane of the sperm egg fuses. Okay, they fuse together. Then on 5, sperm nucleus enters the egg. Okay, when it enters, so there are so many sperms around this. Okay, which comes into contact with the coronary radiator. Some of them dies off before even reaching the zona pellucida, and some of them can penetrate in the zona pellucida. Then the acrosome from the spermatozoa reacts with the zona pellucida to dissolve this membrane so that it enters. Okay, when it enters as a pronucleus, it fuses. It had 23 chromosomes. Now it meets up with the 23 chromosomes from the female, fused together to form 46. Okay, so That's what that's what happens. Okay, so you have to know this. Then what is the site of fertilization? Now we are talking about the breaks in the human fertilization and the human in the woman. Okay, the woman, where does fertilization takes place? So fertilization of the ovum occurs in the ampulla. Okay, ampulla of the uterine tube, just after the ovary. Yeah, the ovary is there, just a small distance from the ovaries. That's where the site of fertilization is. Okay, so what are the stages of the fertilization? So we can just put them in 3 stages approximately. Okay, so there is approximation of gametes. This is the whereby gametes are traveling. Okay, let's say they are traveling because they use it follows by the contraction and relaxation of the female productive system. Okay, so the gametes move by, like, for in terms of the sperm and the egg, they move by peristalsis. Okay, they move by peristalsis, then they meet up. That's what we call approximation, just the travel of gametes in the reproductive system of the female. Okay, and then the second is contact and fusion of gametes. When they form contact, the zona pellucida dissolves, they get to contact, so they fuse together. Now, what after they fuse, what happens next? It's what you're thinking is result of fertilization, which is the formation of a zygote, which is a diploid zygote. Okay, so those are the 3 stages of fertilization, which we can talk about. We can talk about approximation of gametes, contact and fusion, and fertilization. So this is what happens exactly. Okay, so I've talked about approximation of gametes, just the transport of male and female gametes in the female genital tract. Okay, I've talked about that. The spermatozoon transporting the following events play an important role in successful reproduction. Okay, so then we have to talk about, because there is a difference between a sperm and a semen. Okay, so the sperm is the active spermatozoa. Okay, and then the semen is the combination of seminal fluid, organic fluid, and also contains a spermatozoa. Like, we can talk of about 4 substances, which is the seminal fluid, which is secreted from the seminal vesicle, and it makes about 60%, the prostate fluid about 25%, and the bulbourethral glands fluid about 5%, and then the spermatozoa is just 10%, which is the actual sperm we talk about. Okay, yeah, so that's what happens. Okay, so excretion from the seminal fluid are rich in fructose. Okay, so they're rich in fructose, which is a, a which is a monosaccharide. Okay, so this provides energy for the spermatozoa. So energy that's obtained from the spermatozoa, it comes from the fructose. Okay, fructose. Yeah, so that's fructose. And then we can talk about also like how many meals are released per ejaculation, per normal ejaculation. It's 100 million per meal. The spermatozoa at least it's like 100 million per meal of the sperm, and the pH is between 7.2 to 7.6. So basically it's not neutral but alkaline. Okay, it's alkaline, it's slightly alkaline. Okay, like sperm is at least slightly alkaline. Yeah, so site of fertilization, as I said earlier, is the ampulla. It's the ampulla, as you can see here. That's the ampulla side. Then they form an airy cleavage. The egg cleavage now forms a blastocyte. The blastocyte, yeah, it's just the first cells which are dividing specifically around 4 to 6 cells. Yeah, so that forms a blastocyte. So we've talked about that. So now, how does maturity and motility of sperm comes into play? So as they pass through, the sperm which is ejaculated is not actually that mature to fertilize an egg. So it has to take some time to pass through the passage of the female genital tract to get mature. That's what we're saying about approximation of the sperm cell. Okay, so that's what we said about it. So fate of spermatozoa in the female genital tract. So around like 100, 200 to 500 million sperms are deposited in the female genital tract, and about 300 to 500, we're talking about 100 spermatozoa only reach the site of fertilization. So the lifespan of a viable spermatozoa after ejaculation is just the minimum is 24 hours and the maximum is 48 hours. Okay, these sperms, they have a great motility. Okay, so they lose fertilizing power. Then when the sperms, you can talk of the semen in this form, the spermatozoa which is in the semen. When it's ejaculated, they have high motility, high speed, so they get like to get tired fast. Okay, we can say get tired. Let's imagine like it's a person. Okay, so they get tired fast, and then they lose because they were so hyperactive. So before they can even fertilize the egg, boom, they get tired. Okay, so they need approximation, which is helped by the female genital tract to transport that sperm to the site of fertilization. And the sperm, the genital tract of a female is so vast. So these sperms, they attracted to the female ovum or the oocyte through the process which we call chemostasis. Okay, the chemicals which are secreted by the oocytes, they attract the sperm toward them, and that process is what we call chemostasis. Okay, yeah, so that's what they are saying there. Okay, so that's what happens. Now, what is capacitation? When we talk of, we've talked about approximation, the traveling. Now, what's about capacitation? So capacitation is the final step in the maturation of the spermatozoon. Before actual fertilization takes place in the female genital tract, the sperm needs to be capacitated, to be given capacity. Think of this as giving capacity to the sperm cell. Okay, so that's what we talk about. That's what we mean when we talk about capacitation gym. Okay, just giving you capacity to do something. Okay, they give you a go ahead. Okay, you can think of that. Okay, so that's what we mean about capacitation. So certain major changes in the surface of glycoprotein removal. Yeah, this glycoprotein, talking about the zona pellucida. Okay, the coronary radiator is contain glycoproteins which are removed for the sperm to bind. Okay, yeah, so that's what we mean. So like now we can talk of the ovum transport. So the structure of the ovum at ovulation, transport of the ovum from the ovary to the ampulla, the uterine tube, and the variety of the sperm are important for success of the fertilization. Okay, so the structure of the ovum at ovulation contains secondary oocyte with 23 chromosomes. We've talked about that. Okay, so the oocyte is surrounded by zona pellucida, which with proteins, and the coronary radiator. The coronary radiator is the first membrane, followed by the zona pellucida. Then we have the cell membrane, where the sperm enters to meet with the nucleus of the female oocyte to cause fertilization. Okay, so we've talked about this. we talked about contact and fusion of gametes. So we said there are three barriers which the sperm has to penetrate before fusing with the ovum. So these are colonal radiator, that's the first one. The zona pellucida is the difficult one, okay, whereby it has to be it has to be diminished, like it has to be pricked, it has to reduce so that the sperm can enter. And the vetrine membrane is the third one. So that's what we mean about it. So when the spermatozoa attaches to the zona pellucida, when this zona pellucida diffuses and one sperm enters, there is the zona pellucida reacts whereby another sperm cannot enter. Okay, when one sperm fertilizes, the other sperm cannot fertilize. So that prevents forming of a lot of uh, like multiple fertilizations in one nucleus. So it's prevented by the zona pellucida. Then after that, the spermatozoa enters the vetrine membrane, which is just like a cell. We can think of like a plasma membrane. Okay, so that's what happens. Okay, so the acrosome reaction, this is the process of multiple contact that capacitated sperm fate established between plasma membrane and the outer membrane of the acrosome cap. So this results in discharging of chemical substances that facilitate penetration of barriers around the oocyte. Okay, so that's what we mean. So it's covering of the acrosome. The acrosome is the spermatozoa part. Okay, that is removed when it wants to react with the zona pellucida. Okay, it's this membrane, this covering is removed. Okay, so that the spermatozoa can react with the zona pellucida and fuse the glycoproteins which are found there and enters the vetrine membrane, so that this pronucleus diffuses with the nucleus of the membrane to form a zygote. Okay, so that's what happens. So here they are talking about disintegration of barriers, which I've talked about, the coronary jetta. So penetration of this first barrier depends on the release of hydrogenase, okay, from hydronic. So hydrogenase from the acrosome of the sperm is released to diffuse to make sure that this barrier is penetrated. Okay, then followed by the zona pellucida, which contains the glycoproteins, which are ZP3 and ZP2 receptors. Okay, these they are binding sites for the spermatozoa. Okay, to diffuse this membrane. Okay, so that's what happens. Okay, so this is fertilization. The coronal is showing the sperms outside. They're trying to come in to come and diffuse the coronal radiator. Then afterward, they can now go into diffuse the zona pellucida to make sure to disintegrate it. Okay, and enter the cytoplasm through the plasma membrane and meet up with the nucleus. Okay, so that's what happens. Okay, so the vetrine membrane, when a spermatozoa comes in contact with the oocyte, plasma membrane of the two cells fuses. Okay, the plasma membrane from the talking about the spermatozoa and the oocyte, they fuse together. Okay, yeah, that's what we mean about that. Okay, so this probably occurs at receptor sites, the ZP3 and ZP2 we've talked about, that are specific for this species. Okay, yeah, so that's what we mean. Okay, so what's calcium wave in the oocyte? So the contact of the sperm with the vetrine membrane of the oocyte triggers a calcium wave. A calcium wave will mean that the cell is depolarized. Okay, depolarization takes place so that there is that excitation, okay, in the oocyte cytoplasm to come into contact with the spermatozoa. Okay, So then I talked about polyspermy. I talked about multiple fertilization. In this term, we call it polyspermy. Okay, so when the zona pellucida, the ventral membrane is penetrated, these blocks other sperms from fertilizing the egg, and that's what we call to prevent polyspermy. Okay, so there are cortical granules which are produced that prevents polyspermy. Okay, so there are so many processes which occurs there to prevent polyspermy. Polyspermy can result in formation of a zygote with a lot of nucleus, and polyspermy can result in whereby there are deformations to the zygote. Okay, the zygote can have maybe two heads fused, can have 4 legs, 3 legs, yeah, so they can form deformation. So that's the importance of preventing polyspermy, because you only have one zygote which will form. Now if polyspermy was to occur, then the zygote will have deformations. Okay, it can have a lot of maybe fingers when it's growing. It can have fused two eggs fused together. Maybe you have seen all those things happening. So that's the importance of preventing polyspermy. But we don't mean that preventing polyspermy cannot prevent that from occurring. There is a cell division. Abnormal cell division can also cause deformation of the zygote. Okay, the normal formation of the zygote. So that's what happens. So we've talked about the nuclear fusion. Well, yeah, we've talked about this. So this is what happens when we talk about the whole process of fertilization. The nuclear fusion, both head and tail, the spermatozoa comes into contact. Okay, so after they come into contact, the ovum is now called the zygote. Okay, the ovum, in this term we're saying the ovum becomes the zygote because the sperm enters in the nucleus. So the sperm is too small, it enters the nucleus. So we overly refer to the ovum as a zygote. Okay, so that's what happens. So now I said, after we've talked about the process of fertilization, now what are the effects of fertilization, the results of fertilization? So there is completion of secondary meiotic division, the female genital tract. There is a restoration of the diploid number to 46 chromosome, whereby there were 23 from the male, 23 from the female. They come together, they were haploid, now they form a diploid, a complete chromosome, which is the normal number of chromosomes in the human being. Okay, now after that, the effect of fertilization, after completion of second meiotic division, restoration of diploid number, there is also determination of chromosome sex. Now you can tell if that zygote will become a female or a male. Okay, that is determined as the effect of fertilization. Okay, so that's what are some of the effects of fertilization which we've talked about. Okay, so sex determination. So sex determination is crucial. So sex determination is determined by the male. Okay, the male parent. It's not determined by the female, because the female has just XX. Now for the male are the ones who have XY chromosomes. So the ova contains about 22 plus an X chromosome. That makes 23. Okay, now that's for a female. It's just constant 23 plus X forming 23 chromosomes. Now for a male, we have it's either you can release 22 plus Y or 22 plus X chromosomes. So those are chromosomes which contains X are what we call the X-bearing chromosomes, and then those containing Y are what we call the Y chromosomes, the Y-bearing chromosomes. So when since the woman is constant, is 22 plus X. Now you are 22 plus, you are either 22 plus Y or 22 plus X. So when your 22 plus X fuses with the female 22 plus X, that zygote results in being a female. But When the 22 plus X from the female and your 22 plus Y fuses together, they form 46 chromosome with a XY. That now becomes a male zygote. Okay, so that's what happens. Okay, so I just wanted to share with you guys how fertilization occurs and the results of fertilization. Don't forget to like, subscribe, and share to my YouTube channel. And thank you so much for me.